Welcome to the Better Health Guy podcast, where we take a deep dive into complex chronic conditions and explore ways to build resilience, improve vitality, and achieve better health. To stay updated on Scott's work, you can sign up for his newsletter in the show notes, where you'll also find ways to support the show and help make more episodes like this one possible. If you find the podcast valuable, please subscribe, rate, or leave a review, as it helps the show reach additional listeners exploring similar questions. The content of this show is for informational purposes only and is not intended to diagnose, treat, or cure any illness or medical condition. Nothing discussed on this podcast should be considered medical advice. As always, please discuss any health-related decisions with your own personal medical authority. And now, here's Scott, your Better Health Guy.
Welcome to episode 233 of the Better Health Guy podcast. If you are seeking new ways to find better health, I might just be your guy. Today we explore born-free rethinking chronic illness through the lens of systems biology. My guest today is Joshua Leisk. Joshua is a biological engineer and independent researcher focused on complex chronic illness. He's the creator of Born-Free, a patient-led community research project that develops models and potential interventions for conditions such as ME/CFS and long COVID. His work takes a systems biology, reverse engineering approach, exploring how disruptions in metabolism, mitochondrial function, and cellular signaling may contribute to chronic disease. He is known for developing detailed, evolving models that aim to explain how illness begins, progresses, and may potentially be reversed. Joshua collaborates with a global community of patients, practitioners, and researchers sharing pathway diagrams, hypotheses, and research intended to advance the understanding of complex chronic conditions. He is a founder of the Collaborative Sciences Institute, which provides systems biology education for clinicians treating complex chronic disease. He's also involved with Renegade Research, a 501c3 nonprofit, bringing together patients, caregivers, scientists, and clinicians to drive innovation in healthcare. In part one of our discussion here in episode 233, we talk about the many factors that drive these complex conditions. We talk about sympathetic overdrive, the microbiome, we talk about testing, and we talk about the diet. And then in part two of our discussion in episode 234, we'll talk about the immune system, structural issues, hypoxia, oxidative stress and inflammation, the mitochondria, mast cell activation syndrome, and the Born-Free protocol. This conversation was so full of powerful information that we had to make it two parts. The discussion goes deep, so please keep your hands, arms, and legs inside the vehicle and remain seated at all times. I hope you enjoyed this interview as much as I did. And now part one of my interview with Joshua Leisk. When I was diagnosed with chronic fatigue syndrome, before we even called it ME/CFS, very little was understood about the mechanisms that underlie the condition. Today's guest has deeply explored those mechanisms and put together a really detailed model for recovering from ME/CFS and many other complex chronic conditions. If you've looked into his work, you've probably seen his detailed pathway diagrams. Our goal today is really to try to explore the higher level concepts of his model and view chronic illness through his lens. Thanks for being here today, Joshua.
Well, thank you very much, Scott.
So talk to us first a little bit about how exploring chronic illness to the depth that you do became your passion, the focus of your work. Did you have your own personal health journey with a complex chronic illness that was kind of the catalyst for the path or maybe the pathway that you ultimately have taken in life? And then tell us a little bit what's behind the name Born-Free.
Sure. Well, look, I had severe ME/CFS, not that it was called that back in my teens. I was bedbound. At my worst, rolling over in bed was enough to knock me out again for another day. People would carry me to the bathroom, that kind of thing. It was, not a pleasant time. I recovered from that. That was a very slow grind, and then ultimately had a successful career. I sold up the IT business back when I was 38, and at that point, I wasn't quite sure what to do with life. I'd put my feet up for a little while. I traveled a little, And I got focused on doing some metabolic coaching and fitness, and that was kind of a passion for a little while. And then during that time, I had a interesting little moment where I noticed that there were different patterns between some of the clients that I was actually coaching. And some people would respond very directly to normal supplements. And then there was another group that was almost like aliens that would respond completely differently. And that was intriguing. And so I started scratching at that itch and exploring and pulling on that string. And the more that I pulled on that string, the more it kept giving and giving and giving. And the patterns I noticed sort of started converging around certain pathway, impairments and things. And I started looking into nitrogen metabolism and mitochondrial dysfunction. And at that point, I realized that I was looking at my old nemesis again, that this was actually chronic fatigue syndrome. And I found that fascinating. I thought, well, I've got, nothing better to do with my time. This seems like a valuable use of it. So let's focus on this for a while and see where this goes. That was, wow. Yeah. So that was, 6 years ago. And a lot has, has occurred in that time. The understanding has grown dramatically in that time. We've had quite a range of successes, and yeah, we're at a point now where it's something that needs to be scaled.
So where did the term born free come into all of this?
Well, the context for that was the idea that we're born into this world free of encumberments. And that's maybe perhaps not true because we do inherit a number of things from both parents and the environment. But it's also suggestive that if we are able to shed those encumberments, that we can actually revert back to this original state.
So your work focuses on ME/CFS, but there's an extensive list of conditions on your website that you talk about where the protocol may also apply. So we're thinking here about things like long COVID, SIRS, fibromyalgia, Ehlers-Danlos, tinnitus, mast cell activation, POTS, other dysautonomias, SIBO, MS, polycystic ovarian syndrome, Hashimoto's, Guillain-Barré, autism, Parkinson's. It's a lengthy list. And so I'm wondering from your lens, does that mean that these conditions have the same underlying mechanisms in terms of what's driving the conditions and maybe also a similar path out of them? Are they essentially catabolic processes? And are there conditions where the pathophysiology, as you see it, is entirely different? So, what about Alzheimer's or ALS or cancer or maybe aging, which I know you've suggested ME/CFS is essentially advanced aging? Do those conditions have different mechanisms?
Well, that's a very good question. Okay, so in my modeling, there is a common cascade, and then there are individual variables that control the presentation. So in the context, there's mitochondrial dysfunction sitting in many ways at one of the major intersections in all of this. And then there is a localized plus systemic set of impairments. So in that context, the localized section is likely going to be a localized infection. So there'll be particular pathogens involved in a particular tissue. That's going to create a certain personality. The status of the minerals and the heavy metals are going to control a significant amount of the metabolism. And then essentially the presentation is going to relate to the affected metabolism in that tissue. And then ultimately the layers of compensation trying to restore homeostasis in a system that's basically digging a deeper hole every day. So in that context, you can look at any number of those different disorders and diseases and say, okay, well, what does the data show? So you'll see, and the conditions and the disorders and syndromes that I've mentioned in the protocol are only ones that I've seen data for. So it's entirely possible that there's going to be a larger array. It's just that I haven't been exposed to data that actually, allows me to make any kind of suggestion that this is going to be present. And essentially what I've done in that situation is seen the data, done a literature review of that particular condition, found matching, metabolic pathway impairments, and overall suggestions that this is going to be the right area to explore. Similarly, in each of those conditions or disorders, I've seen people putting the protocol or elements of the protocol into play and seeing a therapeutic benefit. So anecdotally, it appears to be the right direction, but further work is needed. We're in the process of moving out to clinical trials for a number of these things, and that's basically where things are at now. So it's early days, but exciting. Alzheimer's, you mentioned, probably a good one I'd mention there. There was an animal model recently. They found that if they restored the NAD redox in that animal model, it reversed the entire trajectory. So all of the memory deficits, everything was restored in the animal model. And look, they used an exotic compound to do so. However, you'll see in the model that there are a number of things that converge to create that NAD redox issue. Figure 14 in my modeling work, for instance, is a really good example of how I would explain a lot of the mitochondrial dysfunction, at least at that core intersection. And then you see the layers of impact downstream and what that does to the glycogen homeostasis and parasympathetic metabolism, and then ultimately how that leads to a progressive sympathetic excess and overdrive, which then has deleterious function on the minerals and the metals, but ultimately it's where like the gastroparesis, the swallowing, the immune dysregulation, and potentially a lot of the microbiome maladaptive response comes from. Something that I haven't sort of talked about too much in the protocol yet, but I have in places, is the concept. There is a link in some of the phobic section that talks about this. So one of the things with the microbiome that's really interesting is that it can respond to shifts in the nervous system. So there was a paper in there where they looked at a mouse model and they basically triggered some changes in the hypothalamus, and within hours, without any food being introduced, the microbiome was retiled and adapted around the signals being sent from the nervous system. Not fully understood how that signaling takes place, but the hypothesis that I would float is that in the context of the fermentation syndrome that I've seen just vastly across like all of the data that's been collected, is that it appears to be a response to try to restore homeostasis to the sympathetic overdrive. So in the context of excessive adrenergic signaling and that moving downstream to CAMP-PKA, so that's cyclic AMP, protein kinase A, that is actually the intersection where the acetaldehyde excess ultimately triggers a downregulation of CAMP via the endogenous opioid synthesis pathway. So as the acetaldehyde ramps up, it's the same as essentially people self-medicating with alcohol. The body then produces the opioids, and then that downregulates CAMP-PKA and tries to restore the homeostasis. The problem you've got, though, is that the species that are often producing this fermentation product also happen to be opportunistic pathogens. So then, in the context of a compromised system, then lead to ongoing barrier dysfunction, inflammation, and immune response. So this becomes part of the overall cycle of why there is this persistence. But it's not the only one that's there. So we see the same things. If you look at like a Parkinson's patient, you look at Alzheimer's patient, you look at age-related diseases, you'll see the same mechanism. You'll see the same, dysbiosis and alterations. You'll see the same patterns in the mineral dysregulation. You'll see the same heavy metal accumulation. You'll see the same immune compromised state. So all of these are features across the board in all of these different conditions and disorders and diseases. So it's then, basically a way of looking at a complex system that's always trying to heal. It's always trying to restore homeostasis, but it lacks the capacity to do so. So in that context, if you've got a system that is always trying to heal and it can't, that's indicating that there's actually a number of things that could be impacting that. So for example, resource shortages, or there could be a range of different insults or toxins which are overloading the system, which once it's reached a certain threshold, it's just trying to keep its head above water and can't actually escape. So that's kind of where the process in the protocol is to restore the resources, reduce the burdens, and then try to shift the direction of all of the metabolism and trajectory of towards recovery rather than progressive decline. And that's essentially how the protocol is I guess, focused.
So, how different are these conditions in the era of COVID, now long COVID? Do you see that people are sicker today than we were before March of 2020? Do you think that those people that are dealing with long COVID have the potential persistence of SARS-CoV-2? Do you think that it's more of a spike protein issue? Do you think that COVID becomes an unmasker where other things like Lyme or Bartonella or Mycoplasma or Epstein-Barr then are reactivated? How has COVID changed your chronic illness model, if at all? Is it different now, or is it more of the same?
More of the same. What COVID has done is give us volumes of data from unfortunate people that are now suffering. And, the COVID interventions were much the same. So in Australia, we had a totally different experience with COVID because we essentially had shut down borders and we had, roughly 9 months of preparedness to get ready for this open border and quote unquote let her rip, as they called it. So progressively, what we've seen is issues where we have reactivation of the herpes viruses. That's one of the most common things you see. There is a mechanism in the protocol and the model which explains that. as part of the mitochondrial dysfunction and a particular fault sensor. There is a fault sensor called hypoxia-inducible factors, 1-alpha. That's downstream of these enzymes called the prolyl hydroxylases. Those are the actual fault sensor. They're multi-purpose. They're also involved in the collagen synthesis and degradation pathways. So essentially, it appears to be that the collagen synthesis is supposed to be upregulated when there's, certain types of mitochondrial dysfunction. But at the same time as that, it's, one of those things that fails under certain conditions, essentially all the resource shortages. But in the context of where we're at now, post-COVID, if we're calling it that, yeah, look, I've certainly seen a significant uptick in chronic disease and health decline and things like that. So sad to say, but yeah, I don't think we're in anywhere near as good shape as we were pre-COVID. And that continues to be the case. But I don't think it's a one-way street either. I think, based on everything that I've seen, it's a totally reversible situation up to a point where, things get very difficult. You've seen the uptick in cancers. We could have a conversation about what I, hypothesize that cancers actually are. And I can discuss some of the studies around that. But certainly the progressive health decline has accelerated. It looks like the background level of insult has gone up in the last number of years, and people's capacity to deal with that insult has been reduced. So in the context of resilience against threat and insult, yeah, essentially we have a population that is less prepared, for the increased level of insult. It used to be that we'd get a flu, season once a year, and we'd have various things going around. And now we seem to get 3 to 4 cycles a year at least of various different respiratory infections. And during that time, every time the immune system is busy dealing with a new threat, it allows the microbial infections to basically increase the size of their biofilms. So it allows them to increase the size of the reservoirs, which then contribute to the overall load. So, normally if an elderly patient ends up, or even an adult ends up in hospital with a respiratory infection, despite it being a viral infection, they'll be given typically azithromycin or some other antibiotic to prevent secondary bacterial pneumonia. And it's for this very reason. While the immune system is busy dealing with a particular class of pathogen, particularly the viral pathogens, it's less equipped, particularly the innate immune response, to deal with the other classes of pathogens, so the microbes. And so this is where, part of this challenge lies. So the more we can do to reduce the microbial burden and or the viral burden, the less susceptible we are to new threats and ongoing, insults.
So you've touched on some of these already, but I want to talk a little bit kind of at a high level, what factors or contributors drive these conditions? Is it long-term nutrient and mineral deficiency that sets the stage? Is it our toxic environment? Is it the acquisition of these chronic infections and the proliferation of biofilms? What are the factors that are setting the stage for the development of these many different complex, chronic, years or decades long health challenges that people have? And when in our lifetime do those factors or contributors begin to impact us? Does it go all the way back to birth, maybe even before? Talk to us a little bit about that spiral that leads to these complex chronic conditions.
Well, I think there are a number of different things that you've touched on there, and they all kind of have slightly different inputs into this context. So we could have, for example, early childhood and pre, we could be talking about nervous system adaptations. We can then look at burdens and different burdens. So for example, aging, one of the differences that I'd look at with aging versus what we see with, say, long COVID or something else in somebody's teenage years is that we have at that point the proliferation of latent herpes virus infections. So varicella, various other herpes viruses that have, been a slow, perhaps smoldering infection that's taken over all the various tissues progressively. I think varicella is probably more pronounced in that context because its ability to replicate throughout the body seems to be a lot more enhanced than, say, Epstein-Barr or some of the others. And you'll notice that, for example, if you get a varicella reactivation or shingles, you're looking at rashes across large sections of tissue. There was a very interesting study where they were looking at cadavers and they were looking at the different viral infections in different tissues. And yeah, there's an interesting little map around that about what was actually seen. Now, going back to the early childhood, now there was an interesting one. There was one of the things that I would talk about is nervous system dysregulation. And that's a key piece because the autonomic nervous system is a big driver in the disease state in essentially dysregulating or unbalancing the homeostasis, essentially pushing the mitochondria beyond, and of course driving the sympathetic excess and overdrive. And that there's multiple loops. I think it's probably good if I was to clarify. It's easy to think about things in straight lines when looking at causality. In biochemistry, that's really not helpful because these are cycles that are all interconnected. And so throwing out one system and the homeostasis out of one cycle essentially affects the others. And all of these different cycles are trying to restore homeostasis. So when we're looking at the ripple effect from one change in one cycle in one system, it then has layers of compensations and ripples throughout the others. So in that context, we could go back to very early childhood, and we could look at, for example, how the nervous system might respond to different threats. And the nervous system is an interesting one to look at because it's the bridge essentially between our consciousness and the biochemistry and the physiology. So in that context, we have our layer of consciousness. If we want to think about this conceptually, we're kind of a consciousness which is experiencing our body and the outside world through an evolved/hybrid monkey nervous system, which is trained to do a whole range of different things. But essentially, one of those things is to keep us safe from threats. So in that context, the nervous system itself, it's autonomic, parts of it. I mean, that's how we're able to breathe when we're unconscious, when we're asleep, things like that. So it actually has its own level of memory and its own ability to react and respond to things ahead of the prefrontal cortex in our actual, our conscious state. So it's important to remember that if the amygdala, if essentially the nervous system is getting trained to perceive patterns, essentially, electrical signals as threats, it's going to learn to predict those as threats, and it's going to essentially, in some ways, potentially keep the consciousness hostage, in this world that it creates, where essentially everything is a danger, everything is a threat, and it can be a horrible, horrible experience when you're stuck in that state. It's like you don't want to be thinking and feeling that the world, acts like a certain way, but ultimately that's the way it's being presented. And it gets particularly difficult if biological processes are being perceived as threats. So this is where it gets really interesting. So if you then look at the nervous system and say, okay, well, it's a bridge. So it's both something that we have to look at from a biochemical angle, but then also something that we have to look at as patterns and programming. So you may have, for example, a series of different threats and mitochondrial metabolism issues, which then lead to energy issues, which then create metabolic alarms in the central nervous system, which then lead to an unfavorable state, then shaping the patterns which are then set to program the autonomic nervous system. So in that context, things that we would normally perceive as safe or low noise type threats, like a level 1 threat, might then be perceived as a level 10 threat because the metabolism is basically biasing the nervous system to perceive it that way. And that becomes very tricky because essentially the autonomic nervous system is learning by repetition. So the repetition of unsafe, unsafe, unsafe leads to a state of lack of safety. And I think that this is something that is prevalent that you'll see in, for example, the autism spectrum, OCD, ME/CFS, long COVID, everything else is essentially the nervous system being stuck in this state where it's reacting, hyperarousal and hypervigilance around essentially like low-level noise and threats. So part of my modeling actually goes quite some distance to explain how the biochemistry sets up the nervous system to become dysregulated in this way. So the cell danger response, Navio's work is really interesting. In fact, that's something that I've taken a few extra steps further to explain that the dampening signals, for example, to the purinergic signaling, so extracellular ATP. Just to take a step back, people would know that ATP is essentially the quote unquote currency inside the cells. Essentially, the ATP is used to activate a number of pathways to produce a whole range of other cofactors. It's also used for like the pumps in muscles, so the ATPases, which then are pumping through the electrolytes in and out of the cells. But inside the cell, it's a currency. Outside the cell, it's actually an alarm signal. So it lands on certain receptors, P2X, P2Y receptors, and that's a way that a cell can communicate with surrounding cells that there's actually a threat, a trauma, or something, there's an alarm. Now, normally there's a dampening signal to that, which is created by another purine called inosine. Inosine comes out of a pathway downstream of the oxidative pentose phosphate pathway, which is downstream of the glycolysis pathway and a range of things. And what I've seen in the data, this is my data that's I've been collecting via Theriome Aristotle reports, very, very, very useful test, is that if we look at somebody that I would call pre-CFS, so they're typically autism spectrum / ADHD, they haven't reached metabolic collapse yet. This is part of the model. And what I see in that data is that they have inosine levels that might be 400% above normal. Whereas once we get deep into metabolic collapse, the metabolism doesn't support producing inosine, and it's typically low in range or very low, which essentially then allows the cell danger response to become dysregulated and treat level 1 threats as level 10 threats. So that's a big piece of it. And then there's all of the rest of the signaling, essentially once you've lost your parasympathetic signaling and metabolism, in other words, localized energy storage, glycogen, then you are progressively more reliant on systemic energy storage, so lipolysis, BCAAs, beta oxidation, and sympathetic signaling on a scale, essentially depending on the energy demands. So in that context, if we have a look at what we see in, for example, autism in children and things like that, we'll see the microbiome dysbiosis issues. We'll see the elevated oxalates. We'll see quite a range of other, histamine, mast cell activation, Ehlers-Danlos syndrome type metabolism and everything else. So that cluster is kind of what we're focusing on in the disease model, because it essentially is sitting there explaining and showing the mitochondrial dysfunction, but also how some of the nervous system and the neurotransmitter dysregulation occurs. Figure 14 on the Born Free website is probably one that I would start with as a good intersection to looking at all of that. And what it basically shows is that the methylation cycle feeds the glutathione metabolism, so essentially the glutathione synthesis. The glutathione synthesis is then required to feed the glutathione redox. The glutathione redox then controls the NADPH redox. And the NADPH redox is then the partner for a number of important pathways like oxidative PPP. So essentially, if the methylation is poor, if you've got MTHFR mutations, that could be, something that predisposes you towards it. If you've got low zinc, and that's progressively one of the things that we see in the model, then that leads you to impaired methylation. That means that you have, and transsulfuration, that means you have low glycine and or cysteine output. And then what that means is that the cysteine and glycine being low means that you aren't producing glutathione in response to oxidative stress generation in the mitochondria. So the glutathione synthesis and redox is essentially one of the automatic cooling systems in my modeling for the mitochondrial metabolism. So as soon as the mitochondria are generating too much heat, so in other words, reactive oxygen species, that causes complex 1 inhibition. So complex 1 is NADH dehydrogenase. It's the enzyme that essentially feeds the electrons from NADH into the electron transport chain, returning NAD. So if complex 1 is inhibited, the NADH pool builds up, and it means that the NADH elevation drives a number of other enzymes that aren't complex 1, which is glutamate dehydrogenase and it's NADP transhydrogenase. So in essence, what happens is that the elevation inside the cell of the glutamate and the elevation of the NADPH pool drives the glutathione synthesis and the glutathione redox, or it's supposed to. However, if there are problems with the glycine and cysteine levels, then essentially the glutamate stays high. The cooling system doesn't work very well. And also, if there's problems with NADPH pool, or there's heavy metals accumulated because the previous low levels of glutathione have allowed the heavy metals to accumulate inside the cell, then they inhibit the glutathione peroxidase activity. So that's the main enzyme that deals with the oxidative stress. And the other heavy metals inhibit the glutathione reductase activity, which means that we haven't got that redox back and forth to maintain the pool. So this combination of problems essentially leads to impaired cooling system for the mitochondria. The engines are overheating. We're accumulating heavy metals, and our ability to detox mycotoxins and every other, environmental insult becomes progressively impaired. At the same time, that affects our glycogen synthesis, our inosine synthesis, our NAD synthesis. All of the key synthesis pathways that we need for energy, for immune, for everything else are progressively impaired downstream of this particular cascade. So this then leads us into a progressive sympathetic overdrive, immunocompromised state, and progressive disease or passenger accumulation as a result of all of this impaired metabolism. And the consequential state of that is that the nervous system cells at a biochemical level are primed towards, progressive threat perception or misperception or categorization.
We'll come back to more conversation around the limbic system, the vagus tonification, the nervous system. But one of the things my mentor, Dr. Neil Nathan, often says is, and I think this is empowering for people, is we don't necessarily need to feel safe in our world and in our bodies, but we just need to feel safer in order to start to regain some of that homeostasis. You mentioned low zinc in Dr. Klinghart's world. That also correlates to cryptopyroluria or KPU, which I'm sure you're familiar with as well. So I'm curious, when you think of something like varicella, the herpes virus that causes chickenpox or shingles, do you think that it is that that virus is really so aggressive in what it does in the body, where it's really the bug that's causing the problem? Or is it the host response to the bug? Is it that we need to modulate our response, that we need to have more tolerance and integration within our own pathobiome?
Ooh, kind of. Okay, that's a tricky one because the answer to both is kind of yes. One of the interesting things, okay, so the viral reactivation is actually triggered by the mitochondrial dysfunction. So chronic viral reactivation. Every time the mitochondrial fault sensors, so the prolyl hydroxylases are inhibited, in that cell, if that cell happens to be infected with one of like the latent lytic viruses, one of the herpes viruses, HPV, HIV, and various others, then that is actually one of the things that can trigger the reactivation. So somebody that has chronic herpes virus outbreak, somebody that has chronic EBV, chronic CMV, chronic whatever, is likely to find that they've actually got chronic prolyl hydroxylase inhibition. They have a bias towards mitochondrial dysfunction. So without the virus or the viruses, that particular fault sensor triggers the anaerobic glycolysis. It triggers the lactic acid metabolism, which then allows a cell to stay alive, even though the mitochondrial function is actually inhibited. So the prolyl hydroxylases, they detect a range of different faults. And then at the point that they inhibited, that stops breaking down the hypoxia-inducible factors 1 alpha, which is the sensor protein, which then triggers the transcription factors to be altered to promote lactate dehydrogenase and to promote pyruvate dehydrogenase kinase. So that has the effect of essentially diverting the pyruvate. Instead of that heading towards acetyl-CoA and then the citric acid cycle, it diverts it towards lactate. So in essence, what it does is that it will allow the muscles and the brain, things like that, to essentially produce lactate. That then gets sent to the liver. The liver then participates, hopefully, in the Cori cycle and then delivers glucose back to the cells. Meanwhile, keeping up the NAD redox and other things. So the glycolysis pathway outside the mitochondria, it's now responsible for the ATP production and the magnesium and phosphate uptake and everything else, while the mitochondria offline, but it's then participating in that via that lactate metabolism. So at the same time that that happens, if the cell is infected with a latent infection, it's likely to now be a lytic infection. Simultaneously, if that, depending on some of the other states of the cell, it's now producing oxalate. So the hyperoxaluria is the other thing that's directly related to this mitochondrial dysfunction signaling. So essentially, if the lactate dehydrogenase is elevated, it biases the glyoxylate metabolism towards oxalate instead of glycine. And that glycine pool then also then sort of feeds into this, same problem with the cooling system, like the glutathione synthesis and redox. So it's a really neat couple of loops which are then impacted once the mitochondria are actually impaired. And so this is where we then go back to, well, is it the mitochondria itself? Is it the TCA cycle reactions? Yes. Is it the substrate pathways? Like, is it the glycogen? Is it the fatty acid oxidation? In transport, is it the BCAAs? Like, where is where are the limitations that are leading to the lactic acid metabolism? Is it the hypoxia? Are we not transporting enough oxygen? Have we got cardiovascular issues? Yes, 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 and yes. So progressively, what you see is the host loses its ability to maintain normal homeostasis. All of these conditions become true progressively. So the heavy metals build up, the magnesium gets low. We end up with low iron and sulfur, which are affecting the iron sulfur clusters in the mitochondria. We end up with the low magnesium and phosphate affecting the glucose uptake and the glycolysis pathway. We have problems progressively with the redox factors, so NAD and FAD and thiamine pyrophosphate and things like that, which are going to affect the fatty acid oxidation pathway. But what's really interesting in all of this too is that the NAD redox appears to actually control the substrate pathway flux. So progressively, as the NAD redox is inhibited, it'll then inhibit via different mechanisms the glycolysis pathway. It'll inhibit the carnitine synthesis, which then affects the fatty acid transport. It'll affect the branched chain amino acids by inhibiting the first reaction and others. So basically, it's signaling to the upstream pathways to slow down. Hey, the mitochondria are overheating, or they're otherwise impaired. Stop feeding us fuel because we're only going to blow up, and take out the rest of the cell.
That's a lot of things to think about there. So one of the things I think many people see elevation of oxalic acid or oxalates, and they think that it's primarily from diet. I know in the mold illness realm, we talk about fungal colonization and organisms potentially producing it. But I think you just highlighted that it could be that diet is actually a small component of the oxalate burden, and that it's actually more the endogenous production that is more significant.
Exactly. Well, I'd liken it to if you're treading water and drowning, someone throwing a bucket of water on your head's not really helpful. So that's kind of the context. The problem is, though, if you then go and restrict the dietary oxalates too much, you lose the Oxalobacter formigenes species, which then means that you interrupt the signaling to then essentially dump and metabolize the oxalates through the gut. So you then progress more towards the kidney stones and everything else, because the urinary excretion, which is going to be enhanced by acidemia, is essentially altered by the lack of the species in the gut. So the dysbiosis progresses. So it's important not to completely eliminate the oxalates from the diet. But yes.
So, from your perspective, how much of these complex chronic conditions is genetic versus many different factors that epigenetically impact gene expression? So, we know genes don't change that rapidly, but it seems that chronic illness is exponentially increasing. So, is the problem our genetics? Or is it the dirty fishbowl in which we live or try to live today?
Well, I think I would answer that in a couple of ways. Most people have experienced life for a number of years in the early days without disease. So at that point, the code is performing really well, right? The programming of the cells is optimal enough to experience a normal, healthy, happy life. And then progressively, that becomes problematic. Epigenetics falls underneath the category of, and if you like, homeostasis, long-term adaptations to try to maintain homeostasis. So somebody that, for example, is an athlete is likely to have epigenetic adaptations towards things that are helpful for fitness. You might have environmental toxins and problems and things like that. Epigenetically, you might then adapt to that environment to then be more resilient to those toxins. So in that context, I think it's probably important to think about the epigenetics as something that, much like the autoantibodies, as something that ultimately is geared towards restoring homeostasis rather than a threat. It's not necessarily, a problem. I think there might be some arguments on the other side of the coin, but ultimately, yeah, everything that I've seen is that the system itself is not stupid and is not trying to harm itself. So it is always trying to restore that homeostasis and adapt to the best state that it can under that environment. So in the context of what we see, like, is the environment becoming more toxic? Yes. Microplastics, really poor food systems and things like that. We could have a half-hour rant just on what happens with the food system now. The environment that we actually evolved in is no longer the environment that we're living. We've tried to make everything more convenient, and we've divorced ourselves and disconnected ourselves from many of the cycles that the environment actually, provides to actually keep us healthy. So the microbiome, the loss of diversity, in fact, there was another interesting study that just came out of Finland. They replaced the rubber playground flooring with dirt, and the immune systems and everything else of the children just bloomed, showing that essentially the microbiome the dirt is essentially a large part of our ability to maintain a microbiome diversity. And probably the reason that I would suggest that is the case is that I think the microbiome is probably best viewed as software updates. It's a biomass. It performs pretty much like the liver, except it can produce probably 1,000 times more compounds than our own liver, depending on whose work you read. And essentially, it's something that is actually interchangeable. So you can update the microbiome. You can be exposed to other microbes, and then the microbiome can proliferate or bloom certain species and respond to host signaling when there's actually demand, in the same way that the liver does. So it's like Windows updates to a base operating system, if you'd like to think of it in that way. So the more that we live, in an environment where we're eating cooked food, where it's all processed, where we're further and further away from dirt, essentially, then yeah, the more difficult it is to actually maintain a healthy microbiome. And some people would say, yes, but there's also all the pathogens and things in the dirt. Yes, there certainly are, but I mean, there's a balance that's being kept. I've said before that if I was ever to suffer a situation where I needed surgery, I'd much rather that I was in a field tent in a forest or an open field than in a hospital, because the microbes that I'd be exposed to during that surgery would be a better balance than essentially the superbugs which have evaded all of the different efforts towards eradicating them inside the hospital. Now, the other side of that is also the mineral content. We've gone to the point where essentially we have a food system where it's actually part of a larger system, where essentially we grow rapidly. We pour all these fertilizers on these different vegetables. They're either fed directly to us, or they go through animals and things, and we're eating the animals. And then the process is essentially drawing the minerals out of the soil, and then we consume those products. And then someone had a great idea that we should then, if we're living in coastal regions, we should then flush those minerals along with all the fertilizer out to sea. So progressively, what we've seen is, since the 1950s, there were some studies on this, that the micronutrient, or the mineral content of the soil, has dropped significantly. It was something like 50 to 60% since the 1950s. So the quality then of the mineral content in the vegetables or the plants, and then everything downstream of that, has been reduced. Conversely, we've seen that the aquatic plants and things like that have had benefited from the fertilizer and the minerals that they've been bestowed, courtesy of our forward thinking. So I mean, it kind of gives us a bit of a nod towards the idea that perhaps we should be looking at perhaps reclaiming some of those sea minerals, and looking at how we would then cultivate and repair the soil. So there's probably a few conversations around that too.
So let's talk a little bit about the testing that you might do to paint the picture of whether or not someone fits into your model and whether or not the protocol might be helpful. So we're going to talk about microbiome testing, organic acids, metal, micronutrient testing, which are some of the key areas that you're exploring. So I'm going to throw out a lot of questions about the microbiome and then let you kind of run. So how do you assess the microbiome, and then talk about the different microbiomes in different parts of the body and how you see them impacting people with chronic illness? So are you testing the sinew biome, the oral biome, obviously the gut biome, maybe other biomes? Is one consistently more important than the other from a health perspective? What are some of those patterns? And then let's talk about the fermenting microbial species that create the acetaldehyde that you mentioned earlier. And since this is really the first kind of dive into that topic, how important is that in your model? What causes elevation of acetaldehyde? Is it the microbes that are in us? You have some amazing tables and things on your website that we'll link to as well, but very, very interesting with all of those things. So I'm interested in, are there some key microbial contributors that you've observed out of that impressive list on your website that are most commonly the drivers of higher levels of acetaldehyde?
Yeah, absolutely. I'll start with how I think the microbiome collapses, and then ultimately the process and the protocol that we're using to address that. So what I think actually happens is that, again, we talked earlier about the sympathetic excess. So basically, potentially that being one of the signals to allow proliferation of the acetaldehyde producing species. There is quite an array of acetaldehyde producing species, and typically you'll find it's the Candida, the E. coli, most of the strep species, staph species, Klebsiella, a range of different fermenting species. There's quite a number of them. Some of them are more prolific than others, and some of them participate as a group. So you might have one species that produces the alcohol, and then the neighbor produces the acetaldehyde, for example. So these things do actually work as clusters. Predominantly what I've seen in the data, though, is that the alcohol doesn't really enter the system much. If we use like an ETG strip, very rarely, you'll see elevations. So it's not like we're actually seeing the alcohol reaching the red blood cells and the other parts of the metabolism to then increase the ETG levels. So it's usually the acetaldehyde directly. What's then interesting is how that happens. So other than signaling perhaps to increase the proliferation, and something just on that, a pattern that I've noticed is, let's say that somebody has, let's say, 14 different species, and it's really common to see quite a range of the overgrowth of the fermenting species. If someone goes through a process to knock out, let's say, 6 out of 14 species that are overgrown, what you typically see is that the remaining species bloom to try to fill the gap. So whether that's just purely a real estate issue because there's space for them to grow, or whether that's because the host is still signaling for their need, and so that's basically driving their growth, that's another question. On the other side of that equation is the traffic cops, if you like, so the butyrate producing species. So something that's come out in more recent data is that a common pattern is the loss of the butyrate producing species, and quite often Akkermansia, which are also required for maintaining the mucin and the barrier function. So in this context, what would happen is if you, let's say that we're in a state of acidemia because of this mitochondrial metabolism issue and essentially this lactic acid elevation and then poor metabolism of that, progressively into more of an acidic state, so pushing towards acidemia, acidosis. One way or another, this is possibly then affecting the microbiome in a way where the pH drop is killing off the butyrate producing species. Quite a number of them are quite pH sensitive. And once the butyrate production is lost, then it does allow for overgrowth of the fermenting species as well. So it's another pattern. So in that context, if you are going to then look downstream of that and also say, okay, well, look, the other thing that metabolizes this acetaldehyde potentially is also some of the other lactic acid bacteria, so the lactobacillus and some of the bifidobacterium. And the other pattern you see is that it's normally a complete or almost complete loss of the lactobacillus and the bifidobacterium. Then progressively, if you're going to try to restore all of this, you could either throw the lactobacillus and bifidobacterium at the system until the cows come home and not see any shift, because essentially the terrain, the environment doesn't support them being able to colonize and then regrow. Or you can go upstream and start dealing with the keystone issues. So in that context, you're not really going to have the ability to restore the butyrate producing species if the system is still in a state of lactic acidemia or acidosis. Progressively, that is harder and harder. So the deeper in the hole you are, the harder it is to potentially restore it. Happily, there is actually a little shortcut to that, which is there is a probiotic species which I think was originally discovered in Japan. It's sold by a company called Pendulum and some others. It's a species of Clostridium. It produces butyrate, but it's actually pH tolerant. So in other words, you can start restoring the butyrate production earlier in the piece while you're still working on the host metabolism. So in the protocol, that's one of the early microbiome interventions is actually to try to restore the Akkermansia and the butyrate production. So the mucin and the butyrate production as keystones before then moving on to the rest of the microbiome. So that's essentially the order. And then of course that has the added benefit once you can then restore the mucin and the rest of the barrier function, then we're dealing with less endotoxin, less LPS, less of the ongoing, sort of systemic inflammatory response. So which goes back to some of the SIRS things. As far as, the microbiomes go, yeah, sure. The bulk of that is the gut microbiome, but it's also important to look at the other microbiomes. We're progressively improving our understanding of the body more as a city for microbes rather than as a single organism. I think the latest data is also showing that we have a microbiome in the brain. Certainly, there are other systems that we see during pregnancy, for example. The gut actually allows a number of the lactobacillus into the bloodstream, hitching a ride on red blood cells, to then end up translocating to the mammary, the glands and the lactiferous ducts, to then end up in the breast milk. And so the immune system doesn't step in and intervene in any of that. It has a relationship with these microbes. So we have that symbiosis. We have all of these other microorganisms inside of our bodies, not just in the gut. I think other than the gut, which is a very, important part of our microbiome because it's the largest biomass, the other important location is the nasopharynx. And that's something that has had a bit of attention, like in some of the studies for the like EAT, so the abrasive therapy, and also you'll see, like if you're in the shoemaker fold, then you'll see basically there's a big focus on mark-ons and what's happening in the nasal microbiome. I think the reason that is so important is because when we start looking again at the parasympathetic and sympathetic signaling, one of the things that's really important is the nerves in the mouth and the nose and how they actually signal the nervous system to respond. So if you are in a situation where we need more energy, so you're breathing more rapidly, breathing through the mouth, you're exercising, that signal sends a need or a signal to the rest of the system, say, hey, actually, we need more adrenaline here. We need more catabolic energy because we have a higher energy demand. The reverse is also true, needs more study. However, deep nasal breathing is actually also going to signal for parasympathetic, so rest and reset and recover. So somebody that has nasal dysbiosis, and it could be overgrowth of fermenting species, which is what we see in marcons and everything else, that inflammation is likely to then also have an effect on the signals, even if we are able to breathe normally through the nose, on the effect of the signaling through the parasympathetic cascade. So in essence, one of the things that has to be done if you're trying to restore glycogen stores and the parasympathetic balance back, from sympathetic bias, is that we need unrestricted nasal breathing and we need a normal, healthy nervous system signaling from the nose to then actually allow that to take place. So somebody that hasn't, dealt with nasal dysbiosis and nasal inflammation is likely to be stuck in a sympathetic bias. So that's one of the reasons that has to be a focus. But the usual story, it's cycles and not a one-way street. So is it again that wherever these microbes are actually residing and like they have their biofilms, they set up residence in different tissues, how do they respond then to the host signaling? So somebody that might have alopecia, for example, somebody that might have, different things, you might have gynae issues, you might have, endometriosis, you might have a range of different things. So if your nervous system is essentially stuck in a state of sympathetic excess, and somehow that signaling for the microbes, wherever they happen to be in the body, to then start proliferating, that then might be actually the signal or the trigger to then allow some of these species, which are opportunistic pathogens, to start creating barrier dysfunction by essentially, proteolysis and things like that. So essentially breaking down our barrier so that they can feed. So that is I think part of the cycle mechanism, how the host metabolism essentially allows for or promotes essentially the growth of some of these opportunistic pathogens that are fermenting species in these different microbiomes. So, and once we end up with the barrier dysfunction, then the invaders are inside the castle. They're basically already in. So now we're dealing with circulating biofilms. There've been some of the recent studies showing that there were like a number of different species, P. gingivalis. There was some strep. I think there was some staph that they found that the atherosclerosis, essentially this calcification. Well, where else do we see these plaques? Oral microbiome. We see oral plaques essentially looking very, very similar to the arterial plaques because they have the same species in them. So once we have barrier dysfunction, then we potentially lead to biofilm circulation, atherosclerosis, and then essentially chronic immune activation because the invaders are already inside the castle walls. So at that point, we're setting the scene to, for progressive chronic innate immune response, which then basically drives a lot of the oxidative stress, which then drives a lot of the immune dysfunction, dysregulation, and essentially presents an ongoing burden because the inflammatory signaling is one of the things that basically drives the mineral transporter alterations. In a chronic inflammatory state, our body intentionally shuts down the mineral transporters in the gut and also signals to sequester the minerals and the metals that are circulating, quite a number of them, there's 11 of them so far in the literature, to store them in the brain, the liver, the kidneys, and the spleen to take them away from the circulating invaders. So basically, it's a gamble that the host is going to be able to deal with the invaders, and it's just sort of locking up all the important resources in some of those tissues while it does so. Makes sense in acute infection, but unfortunately leads to progressive mineral deficiencies in a chronic infection, which hypothetically shouldn't exist if the response to the acute infection was successful.
So I know you have talked about biome site, GI map. Talk to us a little about how you're exploring this Sinu biome. Are you using Microgen DX Sinus Key or using something else for that specifically?
Yeah, the Microgen DX I think is the best option that I've seen so far. So they're a lab in Texas, Lubbock, Texas, I think. And look, they do like it's the usual Illumina and other platform. They'll do similar to doing like a gut microbiome test, but they're quite happy to take specimens from all kinds of different microbiomes and tissues. So in that context, you then get their database of 60,000 plus microorganisms. You get a really rich report on what's actually there. It's a big step up from the typical Marcons testing where they're only testing for a handful of species. So it's you get to see what's good and what's bad essentially. It's not just, oh hey, I've got an overgrowth of staph in the nose, or I've got an overgrowth of candida or whatever. It's like, oh okay, actually, and there's some lactobacillus. You can start looking at the overall picture. That said, typically the profile once you see overgrowth of these things is that it's unfortunately not being balanced by good species in most cases. But there's a very stark difference between healthy controls and essentially chronic disease state.
So, one of the key messages I think that's coming out of your work for me when we talk about sympathetic dominance is many people think of that as stress and working too hard and mental, emotional things, but that there are many physical things happening that keep us in sympathetic dominance. So, even the idea of not getting enough oxygen at night while we're sleeping and someone maybe needing a CPAP could drive a lot of that cascade that you're talking about, right?
Definitely, definitely, definitely. And look, sleep breathing disorder is a huge piece. You would then probably also want to explore, hey, well, what's causing the issues? What's going to actually be creating those obstructions? And then what you might find is some kind of prolapse in that tissue caused by mitochondrial dysfunction, caused by localized pathogens, which then lead to the inability to, essentially activate the muscles in that tissue. So it's quite possible, studies will be needed, but it's quite possible that somebody that deals with the dysbiosis in that tissue might have a reduced or, remove the need for some kind of sleep aid, like a CPAP or something like that. Yeah.
Well, and I think also the occlusion or the bite can also be one of those things. I think people that have CCI or that can also be one of those things that drive sympathetic dominance as well. So, coming back to the acetaldehyde conversation, you mentioned I think it's Clostridia butyric. That's that pendulum product that you talked about. But what are some of the other tools and key strategies for addressing that acetaldehyde issue? Is it more that we need to reduce the production of those aldehydes? Is it more that we need to increase their clearance via ALDH, or is it a combination of both? What are some of the top tools that you're using to kind of clear those aldehydes? And then it sounds like it does also tie back into mineral deficiency as well.
Definitely does. No, you've got it perfectly perfectly right there. So look, it's a combination of all the above. So the host metabolism needs to be able to process the aldehydes. So the essence of that is fixing the NAD redox. So the aldehyde dehydrogenase enzymes, they take NAD, they produce NADH. They're also needing zinc and some other minerals. So progressively, as the zinc and the other mineral status gets lower and lower, then we see progressively the inability to process those aldehydes. So if in the context of a maladaptive microbiome response, it could be that, the level of the candida and the strep and things might be high, but their burden that they're creating is excessive because the host metabolism is not resilient to those aldehydes. So we work on both sides of that in the protocol. The other side of that, though, is that there is a bit of a challenge because as the model and some of the other papers are showing, the issue is that the downstream effects of that acetaldehyde being the endogenous opioid cascade. So if I'm correct in the hypothesis that it's a response to the sympathetic overdrive, one of the things that would be difficult is to then remove the maladaptive, if you like, response to that sympathetic overdrive without having corrected the issues that are creating the sympathetic overdrive in the first place, even if the acetaldehyde is one of those contributors. So if the nervous system slash, the energy systems are stuck in this sympathetic state because that's all that's left, but it's become excessive, then removing the acetaldehyde too early is likely to create more sympathetic excess in essence. So it basically drives the cascade further and further. So in the context of the protocol, we're not actually touching any of that until we've actually started dealing with some of the issues that are affecting the glycogen metabolism and the parasympathetic signaling first. So there is a specific order, and then there's other things that have to happen in there. We can talk about thyroid and a few other things there too. I think the process in actually dealing with that, you need to work on both sides. So work on the host, then work on the microbiome and the adaptations. And I think ultimately the long term, which is why stage 3 of the protocol really tries to clear out some of these biofilms, is that you don't want to be susceptible again to host signaling really allowing a lot of these things to proliferate and bloom. We want to clear out the reservoirs and get a fresh slate.
One of the things that I found interesting, a lot of podcasts here, they talk about this Z Biotics product as kind of a hangover tool, but it actually is part of your protocol for helping to deal with aldehydes.
Yeah, it's one of the interventions. I mean, look, the thing is there are a range of different ways that we can address this. So you can either throw cannon fodder into the system that'll bind to the acetaldehyde and allow it to pass. One of the things that you'll see is like people taking large doses of B1. We also use that in the protocol too. So thiamine is one of the things that will bind to acetaldehyde. And commonly what you'll see is that people might even though they've got a functional thiamine deficiency, they have high levels of thiamine when you actually do serum and other testing. Some of the German labs are pretty good at showing this. So in essence, the hypothesis is that the thiamine is not able to convert into the active form because it's essentially bound to the acetaldehyde. So it shows up because of the way that the lab testing is conducted. It shows as high, but it's not bioavailable. So you can continue to throw, mountains of B1 into the system to try to work around that and also get some functional, thiamine pyrophosphate. But ultimately, there are other ways that we can go about that. So other things that are useful, look, there was the dihydromycetin is another thing that will promote the aldehyde dehydrogenase activity, also used as a hangover cure. It's also then reliant on the NAD redox working. So one of the things that we have to do is really get the NAD redox functioning before any of this aldehyde dehydrogenase activity is really going to be successful. So that, of course, then goes back to restoring the mitochondrial function. And so all of these things, we then start looping and going around again saying, okay, well, to fix one thing, we have to fix another thing. To fix that thing, we have to fix another thing. So we keep going around and around in a circle, and which is one of the reasons in the introduction of the protocol, I basically have drawn some spiral diagrams working or showing each of the different sections and highlights that get progressively worked on in a specific order to keep opening up the metabolism and each section to then progressively allow remediation of each section. So each area of remediation then furthers or benefits the next section. So there's a very, very specific order to these things.
So let's talk then, when we're thinking about the microbiome, how are we improving it? Are we focused on antimicrobials to reduce the pathobiome? Are we focused more on probiotics, prebiotics to really support the microbiome and those beneficial organisms? Are you using antimicrobials first and then rebuilding later, or are you weeding and seeding in parallel? And then how do we durably shift the microbiome in a healthier direction in an era where most adults have little or no detectable bifidobacteria? And how do we do that in a way that it actually takes up residence in the gut and it's not limited to having to take these things forever because they're just moving through but never actually colonizing?
Well, I think there are a few good questions in there. So I think one of the things that I would highlight is that throwing probiotics into a system that's not supporting them is not going to work. They will go straight through and they will not do it. Like you'll get perhaps a benefit while you're taking them, and as soon as you stop, they're not there. So the host itself has to be welcoming. It has to be a welcoming environment for those microorganisms. And it has to be, as I was talking about before with the butyrate and the mucin, we have to actually create an environment which is going to allow these species to flourish. So that is one of the things. As far as antimicrobials go and diet and everything else, these are all, they need to work synergistically, but there is an order to go about the remediation. So for example, if you're going to take a whole bunch of bifido and lacto and you're going to eat all of the GOS and FOS containing foods and things that are actually going to help sustain them, if they have no ability to stick to a surface, you're still going to be feeding them, but they're going to go out. You're not going to retain them. So we go through that process. So it's a matter of getting the barrier function and the mucin sorted out, getting the butyrate production up, and then introducing a mix. It's kind of back and forth. It's not something that we have to do everything all at once, and it probably won't work that well. So it's basically a way of a process of going through and essentially trying to seed the missing species, feed the missing, the new species, and make space for them by removing some of the less helpful species. So in a long enough time span, this is something that will, spin up and correct itself. It's always going to try. But if the foundations aren't there, you're going to be throwing probiotics at the wall, and it's just not going to work.
So before we move on to organic acids testing, I want to talk a little more about the microbiome, specifically on the pathogen side. So in this population that you're working with, how common is it to see Lyme, Borrelia, Bartonella, Babesia? Do you view the viruses as more opportunistic, like many people do? I see a lot of practitioners will say, well, if we just treat the other things, the viruses will resolve themselves when the immune system becomes more balanced. Or do you think they're actually more core drivers of the disease process? And then I'd also love to hear where you think parasites fit in this model, maybe fungal colonization from water damaged building exposure as well. And then how are you using cholesterol as a potential proxy for the presence of chronic infection?
Oh, these are good questions. You've done well there, Scott.
Coming from you, that's incredible. I'm very honored.
All right. Well, look, I might even start with the LDL question first, just because it's a good one. So the LDL, there is a particular enzyme called HMG-CoA reductase. And HMG-CoA reductase is essentially the regulator for the cholesterol synthesis. Now, there's a good paper on this. I think it's linked in the protocol somewhere. But essentially, if ALDH2 is inhibited, it dysregulates the HMG-CoA reductase. So essentially, LDL can be used as a proxy for NAD redox inhibition. And essentially, you can use that then. It's like everything else, there's cycles involved. Like elevated cholesterols might have some downstream effects on certain parts of the metabolism as well. There might be problems with glucose, there might be certain other things. But at the same time, it's also a canary for deeper problems within the rest of the system. So we can also use a range of other things to infer the status of the NAD redox. So elevated B6 is another really good one. Elevated B6 in the serum is another indicator for problems with like the pyridoxal kinase activity, so ATP, MGATP, in fact, more specifically ZNATP, but also the aldehyde dehydrogenase activity that breaks down the pyridoxal into pyridoxic acid. So it's a good proxy. Same with elevated vitamin A precursors. So you have essentially the same metabolism. You have retinol, which is the alcohol as sorts. You have the retinal, the aldehyde, and then retinoic acid, which is all of these are requiring NAD redox and aldehyde or alcohol dehydrogenase type reactions to create them. So if we have an elevated beta carotene, for example, or elevated retinol, we can use that again as a proxy for inhibited aldehyde dehydrogenase or inhibited NAD redox. So these are all good proxies for looking at basically that particular intersection.
So I just wanted to get your thoughts on where or how common Lyme and co-infections are, parasites, fungal colonization, and do you see viruses as more opportunistic or core key drivers?
So yes is the short answer. They're essentially cycles. So they are both a sign of an underlying problem, but they're also something that will create new problems. So they're part of a cascade. They're a series of loops. So with the viruses, as I was talking about before, with the hypoxia-inducible factors, 1 alpha, again, that mitochondrial metabolism, if you sort out the mitochondrial metabolism so that HIF-1 alpha is getting broken down, then at the same time as that, you're going to remove the reactivation of the viruses. So they're no longer triggering inflammatory processes and immune response, which then means the immune system's also free to deal with some of the other threats, which might include, for example, Lyme and parasites and other, infiltration of other species. So fungal infections and tissue infiltration. So basically, again, fixing the mitochondrial dysfunction then allows the immune system to become regulated. And importantly, as soon as you fix the energy systems, that doesn't just benefit muscle and brain, that also, benefits the immune response. So in that context, a buildup of, Bartonella and Babesia and other things is as much a sign of being immunocompromised and poor metabolism, as it is a potential for ongoing, threats. Now, in that context, I'm probably one of the 40% of people potentially in the population that has, Bartonella and Babesia and probably a bunch of other things, and I have no symptoms. But I don't have problems with the mitochondrial metabolism and the other energy systems where essentially they present as a symptom. On paper, I can see lymphopenia and neutropenia at times if I'm really pushing my metabolism in my heavy fitness days. So before I was more, invested in the chronic disease research, I would see if I was overtraining, I would see the lymphocytes and the neutrophils dropping, and for periods of time. And that has been, progressively a thing since I was very young. Like the whole family basically has that. And we thought it was, perhaps genetic. What I've seen using Walter Torello's microscopy and staining is that I have grade 3 Babesia and grade 1 Bartonella. So it's possible that when I had my first response to my first COVID infection, and that knocked me out for quite a while, and then I had poor glycogen storage for about 6 to 8 weeks after that. That I think was probably the proliferation or reactivation, if you want to think of it that way, as some of the circulating species that have been carrying for decades, without any symptoms. And it's the same thing. I think I reactivated Epstein-Barr at that time with that infection, and a few other things too. But it basically just was pointing towards, the host's metabolism versus the landscape, the pathogenic landscape.
So with the fungal colonization, I mean, we know there's things like Candida or Candida, but do you also think that the environment in which we live and work is a potential contributor to fungal colonization in the sinuses, gut, or systemically, things like aspergillosis and other mold type organisms?
Definitely. I mean, you need to be exposed to the species, and the rate of exposure basically controls the rate of colonization or threat that the body has to actually deal with. One of the things that I mentioned in the protocol is that if you are dealing with, constant rhinitis or sinusitis and things like that, it's entirely likely that will have some sort of issue in the home, which is then leading to this chronic threat. So it's a burden to the system. And again, like some people, if you've got functional glutathione metabolism, if you don't have issues with methylation, glutathione, OXPPP axis, for example, you could be exposed to the same level of burden as somebody else that is compromised, and you'd be fine. You might get, some nasal inflammation on occasion or something like that, and otherwise go about a quote unquote normal life. Whereas somebody that is already deep in the hole, the same level of burden just is able to push them into a very unpleasant place. So it's host resilience as much as it is the level of background threat. So anything you can do to improve the burden is going to improve your overall trajectory.
So let's shift now to organic acids or OAT testing. There's many of these that have come up. You have some pretty interesting interpreters on your site for Mosaic, which I think is your kind of preferred organic acids testing. The more recent Theriome Aristotle, which I did and ran through your interpreter, by the way. There's also organic acids testing from Vibrant, Genova, and others. So what are you looking for in organic acids? What are the key patterns? What do you often find? Is that where you're also getting some clues for lactic acid and anaerobic glycolysis? Are you looking at any of the fungal colonization indicators? What are the most common things that you see? And then when people run these through your interpreters, is that then providing dietary and supplement guidance? What are they getting out of the interpretation of that metabolite profile?
Well, these are all good questions. So each of the oats, they have their own strengths and weaknesses. Even the less rich oats, the ones with less markers, are still helpful. So they still cover most of the key intersections, but they don't have the resolution on the pathways that, say, the theriome is going to. So if we really want to have a really good look at glycolysis, for example, then the Theriome Aristotle Plus is going to give us a number of different places in the pathways, and also towards glycogen synthesis and glycocalyx and blah blah blah, that we can actually then go, oh, that's really interesting. It is, I would say, really important to look at that in the context of the ionome as well, which is why we also have the mineral testing. So if we take the metabolites, and we can say, okay, well, this particular, this one's up, this one's low, it's then important to then look at the reactions that create them and say, well, what are their cofactors? And if a particular cofactor needs NAD and it needs magnesium, for example, or something else, and you then look at the cofactors, and you've either got a direct measure, which is really nice with the Aristotle Plus, or you've got an inferred one that's been calculated based on the patterns in the oat markers, and the tool does that. Then yeah, you can basically say, okay, well, that's really interesting. So yeah, I've got a 40% deficiency in magnesium indicated, and I have a deficiency in NAD indicated. So it's no surprise then to see that this particular metabolite is low. Or there's the one, another one might be elevated, and the same sort of thing. The one after that might be inhibited. So it's basically being able to look at multiple layers at one time and see the cause, not just the outcomes in the pathways. Now, once we have that data, then we can start looking at the patterns and the intersections. So one of the things that I highlight is there are a few, if you like, there are a few alarm bells in the dashboard of these organic acids tests. So one of them, for example, you mentioned there was lactic acid and the oxalates. So they're a good indication for the prolyl hydroxylase inhibition. So in essence, if we're seeing the lactic acid to pyruvic acid ratio, that's a good indication that the metabolism is favoring cytosolic NAD redox, in other words, lactate dehydrogenase. But depending on the fitness of the glycolysis pathway and glycogen and diet and things like that, the total for both the lactic acid and the pyruvic acid might be low. So there's not enough substrate there to create vast amounts of lactic acid. So we can use other proxies. We can look at the oxalic acid as another indicator for the same thing. So even if somebody was on a ketogenic diet, poor thyroid, blah, 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 and you'll see that because you might have elevated ketones and acetic acid and things like that, as long as they also have problems with the transamination activity at glyoxylate, you'll see the elevations at oxalate. So we can use both of those as proxies, essentially, for prolyl hydroxylase inhibition and cytosolic NAD redox use. So they're two very useful alarm bells in the dashboard. Now, the other one that I really like is 2-hydroxybutyrate. So 2-hydroxybutyrate is another one that it requires lactate dehydrogenase to produce it. It takes alpha ketobutyrate and then produces 2-hydroxybutyrate using NADH as a cofactor and lactate dehydrogenase, potentially another enzyme, to create it. So that elevation of 2-HB relative to alpha ketobutyrate, where you've got that measure, again, the Aristotle plus, is a really interesting marker that you can use to infer the NAD redox. And it's also then the key, if you like, sensor that is used to then inhibit the branched chain amino acid catabolism. So in other words, if 2-HB is elevated, it inhibits BCAT1 and 2, which then inhibits the branched chain amino acid catabolism at the top of the pathway. So iso, leucine, leucine, and valine, where they get broken down and eventually converted into acetyl-CoA and propionyl-CoA. The other ones that I would look for, the status of the TCA cycle. If we have a look at the other regulator for the fatty acid transport and oxidation, if we look at that pathway, it's fairly short. So we get essentially the protein breakdown. It then goes through a couple of, in the pathway, there are a couple of hydroxylase reactions, very similar cofactors and behavior to the prolyl hydroxylases. So they're sensitive to hypoxia, they're sensitive to oxidative stress. So in other words, they need vitamin C. They have iron, and essentially they will take alpha ketoglutarate, which is one of the metabolites in the TCA cycle, as a substrate, and they will produce succinate as a product. So in other words, if the TCA cycle is being impaired for various reasons, then it means that those two reactions in the carnitine synthesis pathway are going to be expected to be equally impaired, which is then going to produce a lower amount of carnitine. Similarly, there's an ALDH enzyme in that pathway, ALDH 9A1, which essentially is a NAD to NADH plus zinc plus whatever. So progressively, if the NAD redox is inhibited, if we've got problems in the TCA cycle, if there's oxidative stress, if there's hypoxia, the carnitine synthesis is slowed down, which then reduces the fatty acid transport, which then means that there's less substrate being shuttled into the mitochondria to then get oxidized. Now, that's really interesting because, sure, it's regulatory, but it also then has an impact on fatty acid accumulation in the cytosol. So in the context of non-alcoholic, quote unquote, fatty liver disease, it's basically you've got CD36 and others shuttling all these fatty acids from lipolysis, sympathetic overdrive, into the cytosol, but then not getting transported into the mitochondria for oxidation. So that buildup of those fatty acids is essentially where, what we'd be looking at in non-alcoholic fatty liver disease. Of course, according to the model, even though the person is not consuming alcohol, the microbiome producing the alcohol and acetaldehyde essentially creates the same effect. So you end up with the same NAD redox issues, which then leads to the same TCA cycle issues, which then creates the same carnitine shuttle issues. So in other words, it basically is the same, same process. Other things that are really interesting, we can look at the neurotransmitters. So we can look at the synthesis and degradation pathway for the catecholamines and also for the serotonin metabolism, but also the kynurenine pathway. So all of those are important. We can look at, you were mentioning, I think, pyrols and other things before. So if we're having a look at, for example, the dopamine synthesis pathway, there are a number of different things which can go wrong with that. So there's AADC reaction that needs magnesium and P5P. So if we've got problems with magnesium and P5P, we might see a bottleneck there and see a buildup of what might be happening with L-DOPA. If we've got problems with oxidative stress or low zinc, if we've got problems with other microbiome issues, then we might have a deficiency of BH4. So in that context, we need methylation to recycle the BH4. We're going to need the BH4 synthesis pathway, which requires quote unquote vitamin Q or quine, which is normally produced in the microbiome. It also comes from certain dietary items that most people end up avoiding. So wheat, as in wheat germ, and dairy are good sources of quine, and that's then needed to produce quosine, which is then also and part of the BH4 synthesis pathway, but also responsible for some of the mitochondrial oxidative stress issues. So in that context, we might see, an elevation of phenylalanine. If we've got a therium Aristotle plus, we might see tyrosine. I think the OMX also has those. We could look at some of the, the relationships there and see if there's a bottleneck, and that might give us some clues as to what's going on with BH4 and/or iron and/or oxygen. We can then also look at the degradation pathway, which needs to be kept in balance with the synthesis pathway. So we could have a look at and say, okay, well, I've got an elevation of DOPAC, but I have low, like HVA, for example. So that's going to suggest that there is a SAMe/magnesium deficiency affecting COMT. We could see, for example, that we have low DOPAC, and we might have like elevated DOPAL. That's not directly measured yet in most of the oats. But if we see very low DOPAC while we've got elevated L-DOPA/dopamine, then we can probably get a clue, particularly in the pattern of other things that might be present, that we've got a NADH and ALDH issue. So all of those things become present. And then on the other side of the coin, when we're looking at the serotonin or the tryptophan metabolism, we might see, for example, that we have elevated kynurenine pathway elevation. So that's the precursor towards NAD synthesis, which is in competition with the serotonin pathway. So if we have various things which are going to promote the kynurenine pathway towards NAD synthesis, then it's going to affect, if there's low tryptophan availability and other things, the availability of serotonin and then melatonin and other things, which are on the other side of that competition. So, and then, there are various different things that are going to regulate or dysregulate those pathways too. So if we look at the patterns in the oat markers, and particularly with the minerals in combination, we can look at these pathways. We can actually see visually by using the metabolic pathway overlay tool exactly what's going on. So it then allows us to make better clinical decisions about how you would step in and intervene in that situation. So this is where these tools are a totally different way of evaluating and remediating these situations that should be present in clinical care and currently aren't. So that's the focus of my work at the moment, is to introduce them.
I think the other challenge of your work is how to get us mere mortals to understand it. And fortunately, I know you're starting to do more training with practitioners and things in that realm, which will really help, because it is overwhelming, as I'm sure you've heard before, for many of us to try to understand all of this. But you've done an incredible job putting it all together on your website. So before we step away from organic acids, give us a little more insight, so people listening that maybe aren't familiar with aerobic and anaerobic glycolysis, where we see more lactic acid, what are the factors that shift us into that anaerobic glycolysis state and to the production of lactic acid? Does that have then any effect on blood pH, which is generally very tightly controlled? What are the problems that result from that acidosis? And then what do we generally need to be looking at to shift back to aerobic metabolism, where we much more efficiently create ATP and also lower that lactic acidemia? And then coming back to oxalic acid, which you mentioned a minute ago, I think that's also a potential indicator that someone is in that anaerobic glycolysis.
So essentially, the lactic acid metabolism or the anaerobic glycolysis can be triggered by a number of different faults. In the healthy population, under normal circumstances, under exercise conditions, if the oxygen is insufficient, if the glycogen availability inside the muscles insufficient, then and ultimately if the demand for that energy exceeds the ability of the localized energy storage and the systemic energy availability, so like the fatty acids and everything else to deliver that, then what will happen is that the TCA cycle flux will be insufficient, and that's going to trigger the prolyl hydroxylase failure at that point, which then triggers the lactic acid metabolism. So there can be a range of different things. We could have oxygen issues, which are affecting prolyl hydroxylases directly, but also just the respiration required for that pathway. In the chronic disease spectrum, we have different influences. So we're not so much worried about the exercise driving this, because essentially the basal metabolism is already enough to trigger this lactic acid state. So simply getting up out of bed might be enough to trigger a whole range of lactic acid metabolism in a severe state. Or somebody that's housebound, there might be that getting out and walking, 1,000 steps for the day is enough to, put them into a quite a severe state of lactic acidemia. There are a few things to look at there. So in the context of consequences of doing that, and this is one of the key things that the model's describing, is that the more time we spend in this acidic state, the more that we're losing some of the host materials. So the electrolytes get excreted, the zinc gets excreted, and ultimately leads to this progression, this worsening. So this is one of the things that in PEM that I describe as basically why people should be pacing, is because they want to stay out of this lactic acid metabolism and state so that they're able to then replete these resources. So it's a key part.
For people listening, PEM is post-exertional malaise.
Yeah. Figure 12 on the website does a good job, I hope, of explaining this, these cycles. But essentially, it puts mitochondrial dysfunction as the core in the middle of the diagram, and then shows the relationships with the mineral losses, with the hyperoxaluria, with the renal excretion, with the chronic viral reactivation, with the barrier dysfunction, with the hypermobile Ehlers-Danlos syndrome type metabolism and the vascular version, with the lead into the structural issues, the chronic cranial instability, and all the rest of this. So the hypermobile Ehlers-Danlos syndrome type metabolism is described in the disease model as secondary to the mitochondrial dysfunction and the mineral losses. So, conversely, what I've also seen anecdotally, and we still need to go to trial, but the trials, essentially, when you resolve all of this, then those connective tissue disorders are also, removed from the equation as well. So
Amazing.
it's not a one-way street, and it's not I see arguments on Twitter, and it's like talking about, oh, it's genetic. Okay, all right. Well, maybe in some cases, no. You'll see so many people where they've had, like, suddenly they've got long COVID, and then now they've suddenly got all these connective tissue issues, and it's like, well, yeah, I didn't have that before. Now I've got this. Okay, great. Well, let's have a look at the markers, and what do you see? Oh, look at the oxalates, look at all these other stuff, and then now when we look at the Therium Aristotle plus data, look at the proline, look at the hydroxyproline, look at the glycine levels, look at all the things that are required. So it becomes very mechanistic, and the better the data, the easier it is to see all of this and communicate it. And one of the things that I'm really, really thankful for with the Therium data is that it's made my life so much easier to actually describe all of this, particularly with the metabolic pathway overlay tool. It's been a childhood frustration of mine to not be able to communicate the things that I can see and do. Yeah. So there's, having these tools and this data it's a blessing.
So I know Therium just enhanced it with about another 100 markers. Is your interpreter already looking at the latest version? It is. Wow.
It does both.
Incredible. It's funny in this whole conversation, usually when I'm talking to someone, I do this kind of light bulb moment thing, but for you, I'd have to like stand here and just hold it the whole time, because like every other sentence, there's a light bulb moment. So when we're thinking about the TCA cycle, Krebs, energy production, I'm wondering, do you use the Mitome test that we're getting a lot of conversation around recently? And then when we're looking at kind of the NAD side of things, which we'll talk more about later, have you used the US Biotech or Ginfinity NAD panels at all?
I have, and look, honestly, you don't need to. A few things missing. Okay, so when we're looking at the mitochondrial metabolism, just taking one step back, the Therium Aristotle Plus already has NAD, NADH, NADP, NADPH, ATP, and a whole range of others as direct analytes in the panel. So that's one thing. So you don't need to go and use another panel. If you're getting the Therium Aristotle Plus, it's already got it all. You don't need to do the others. The other thing is that one of the diagrams you'll see in the website, when we're looking at the mitochondrial metabolism and the electron transport chain, everyone learns about complex 1, complex 2, complex 3, complex 4, and complex 5. And more often than not, they don't learn about the other feeder enzymes, and you'll see them represented in that diagram. So there'll be things like choline dehydrogenase, as an example. There's GPD1 or 2, which has a position in the intermembrane space. There is all of the other FAD, so the riboflavin related feeders. So there's ETFQO, so the electron transport flavor protein, and oxyreductase, and that's taking like basically all of the FAD-H, which is generated inside the mitochondrial matrix, and potentially feeding that into the electron transport chain as well. You've got the proline dehydrogenase, you've got hydroxyproline dehydrogenase. You've got a range of these other enzymes which are direct electron feeders into the electron transport chain, and they've got to balance. They need to balance essentially with the NADH input, so the complex 1 input, because otherwise the motive force, the flux, the proton equation becomes unbalanced, and that can actually push the electrons back down complex 1 and spray them inside of the mitochondrial matrix. So there needs to be essentially this balance kept between these different feeder enzymes. And unfortunately, that is one of the things that is not so easy if you've got, NAD redox issues at the same time as compensation, elevation via some of these other pathways.
Another one of the tests that you talk about is the Mosaic Tox Detect. There's also the Vibrant Total Toxic Burden Panel. Both of those appear to have this HEMA, or 2-hydroxyethyl mercapturic acid marker, which you also talk about being a potential indicator for acetaldehyde. So I'm curious in general, how environmentally toxic, when you're running these tests, are you seeing people with complex chronic conditions? And could it be that early on in the process, when someone doesn't have the energy currency to really detoxify and their drainage pathways are not supported, that those early tests are kind of tip of the iceberg, and later you actually see much higher levels, even though the person is symptomatically better, they're detoxifying better, they have better mitochondrial function?
You've done your research here, Scott. So yes, short answer, yes. So
I
look,
have to give credit to that one for Neil Nathan, so that's why I
the
learned about the tip of the iceberg thing. So
So one of the things, and look, you see the same thing with heavy metals. So it's the same intersection, same metabolism. We're talking about glutathione synthesis and redox essentially as allowing these things to accumulate. So while we've got impaired NAD redox, we have impaired detoxification. And so that's mycotoxins, that's heavy metals, that's a range of different things. And so in that context, yes, when you look at say like heavy metals in urine and mycotoxins in urine or hair or blood, wherever it happens to be, if they're not getting kicked out of the cells, in other words, if the glutathione, metabolism is impaired, then it actually looks really good. Might actually look like low heavy metals, might look like low mycotoxins. And then as soon as you start fixing methylation and glutathione metabolism, suddenly you see this, enhanced excretion. It goes right up. And people might feel a little bit bad about thinking they're doing the wrong thing. Oh, look, suddenly it's elevated. I'm going, I'm doing worse. I'm feeling great, but I'm doing worse. No, you're actually doing much better at actually chelating and actually getting the heavy metals out, or you're doing better at actually getting the mycotoxins out. So there's going to be a period of enhanced excretion. And of course, you can do things with binders and other things that are going to be helpful in reducing the total load during that time. There's a section about binders in the protocol and kind of a matrix of what they target and don't target. And depending on the data, you can choose, from using that matrix if that's helpful. Sorry, your doctor is able to help guide you on that. And yeah, so that you would expect to see a period of enhanced excretion as you start improving the terrain.
So while we're still on the topic then of environmental toxicity, I want to talk a little more about how you suggest people do the environmental assessment remediation. Are you having people do an ERMI or some other test of their home and workplace as part of the protocol? What do you usually find? And then what does remediation or mitigation look like from your lens? Are you generally having people attempt to remediate the environment they're in, getting to a different environment, cleaning the environment with ozone, which there's pros and cons on that side of things? Does it mean that you start to focus on detoxifying the mycotoxins, addressing the fungal colonization? And then how often do you see someone that is bedridden in the exact environment that's going to keep them bedridden?
Yeah, all the time. So, okay, some good questions there. So, look, you can obviously do ERMI testing. There are problems with ERMI testing as well. It'll give you a bit of an indication. The idea that you need to have a perfect environment to heal is not correct. I actually healed from my own ME/CFS in the same moldy environment that I was sick in. For example, it's not impossible to do that. If you're able to restore the terrain, get the nervous system back under control, all those sorts of things, and particularly get the detox systems working, then you can turn it around. But it depends on, a number of different factors. So of course, anything that you can do to tip the balance in your favor is going to work towards improving your trajectory. So if you can remove the environmental sources of the mycotoxins and continue doing that, because your body is the vehicle in which most of these things are actually carrying between environments. So you can go and move, you can go move to a hotel, you can move house. And what you'll likely find is that, you will actually end up with, ongoing, fungal issues and things like that inside the environment, around the bedrooms, the windows, the blinds, bathrooms, things like that are going to follow you because you essentially are the carrier. There's always going to be a certain amount of environmental, it's like essentially the wind blowing in, and do keep the windows open. It's a big improvement, particularly for sleep and everything else. It's always going to be something that you're exposed to. Like mold is a normal part of the life cycles in the ecosystem. The fungal role, if you like, is to break down an unfit organism into the resources that another organism can reuse. So what we're trying to do is make sure that we're not an unfit organism, so that we're not susceptible to these insults. It's a test, if you like, of fitness.
And when you say you are the carrier, are you meaning that the person has become colonized with the fungal organisms, and as Jill Krista would say, you have become the moldy building?
Yes,
Yeah.
definitely. Yeah. So you can go through and you can clean up, you can move into a brand new pristine environment, and within a short period of time, you'll notice that, oh, where did all this mold come from? You've brought it with you. So at the end of the day, I don't feel that it needs to be absolutely perfect. It just needs to be good enough. It needs to be less of a burden than your system has problems tolerating or is resilient to. So visible mold needs to go. If you've got, things that are affecting the mycotoxins, because most of the mycotoxins are aerosol, so they're gases. So you need to basically clean up as much of that as possible so that you're not having to deal with it.
You also look at blood type, comprehensive blood chemistry, metabolic panel, iron, LDH, lactate dehydrogenase, lipids, B6, and vitamin D. So let's talk about the LDH isoenzymes. I had not heard about that in any detail until looking into your work. I actually have a requisition for a blood draw on Thursday where I've now added that. So we know that LDH is involved in the conversion of pyruvate to lactate, but what are you looking for when you're evaluating those patterns? How does that tie back into that shift between the more efficient aerobic metabolism and towards the glycolytic or anaerobic state, even if oxygen is present? And what are you gleaning from LDH isoenzymes?
Well, the important thing there is that when we're looking at all of these issues, we need to be context specific. What cells are producing the lactic acid? So where's the burden coming from? And then ultimately, how's that being metabolized? So if you're looking at the LDH isoenzymes, we then get a glimmer of an idea about which cells are producing or metabolizing the lactic acid. So the typical pattern will be that you'll see a bump in LD4 and LD5, which might be indicating, for example, that you've got the muscles producing the lactic acid and the liver is likely metabolizing that lactic acid back into glucose. So that's kind of the Cori cycle upregulation, giving you an idea that you're an endurance athlete, even though you might be in bed or housebound. So that's a bit of the pattern. There might be other issues. We might see, some bumps in one or two, for example, and that might indicate some other tissues to have a look at because there might be some vascular issues. There might be some other things that are problematic. There could be like lung dysbiosis slash asthma slash COPD, which are essentially affecting some of the other metabolism. And all of these things have loops. It's interesting because if you look at a standard CMP, they include typically LDH total. And one of the problems with that total is that it can easily mask the individual LDH isoenzyme elevations. And if anyone from New Zealand's health regulatory side is actually listening to this or watching this, may I suggest that you modify your policy of only having LDH total, because it is unfortunately completely insufficient for the task at which it has been given. So the LDH total, you could have, for example, somebody that has an LDH, and it's typically, look, the clues you get are if the LDH is like 160 and up and the person's sedentary, it gives you a bit of a clue that it's quite possible that there's going to be a bump in one of those isoenzymes. But according to the lab range, which is say, oh, well, it might be between 120 and 250 or something, doesn't really tell you that there's a problem. If you are a GP or somebody that's not really, educated on this particular topic, you go, oh, yeah, well, that's in the middle, that's fine. And then if you do the isoenzyme, panel, and you go, oh my God, the LD4 and the LD5 is like through the roof, and everything else is so normal or low that it didn't capture it in the total. So the total gives you a red herring. It gives you a false sense of security about, the anaerobic glycolysis in a specific context or cell types. So that's why it's an important one to use if you're trying to get some sort of indication of what tissues to look at and then why there might be that elevation. So that's why I make a good mention of that in the protocol.
So, one of the things in the past few years that I've probably become much more aware of that I didn't think a lot about early on was the key role of micronutrient deficiency. Minerals, particularly, but also B vitamins being another big one. So, you in your testing also look at the oligoscan. I think some parts of the world call it the VitaScan, also micronutrient testing. So, with the oligoscan, are you seeing patterns of toxicity like high aluminum, or are you seeing more specific vitamin and mineral deficiencies? So are you getting more insight into the toxicity side or deficiency or both? I know that Genova has a NutriVal. Vibrant also has their micronutrient testing. I think they recently changed it to something like the Nutrient Zoomer, if I remember correctly. You talk about the Cell Science System cellular micronutrient assay. So what are the patterns that you see in people with complex chronic health challenges? why are those minerals so key to your model? Are there certain patterns of which minerals are most commonly kind of the priority deficiencies? You've talked already about why they're deficient to begin with food and soil and those kinds of things. And then are there specific B vitamins that you also see commonly deficient that are also key to this whole conversation? And then lastly, because I see this a lot and your work gave me a new insight, is the vast majority of people that I see their labs, they have low white blood cells. And so a lot of times we jump to, oh, that's something we see in Lyme and co-infections or so on. But could that also be a proxy for poor nutrient status?
Yes, and again, very good questions and very well researched. So, if we were looking at the B vitamins, okay, there's a figure on the website. I think it's figure 7, and that talks about essentially the coenzymes, so the active forms of the B vitamins in particular, which are downstream of zinc deficiency and other things. So when we think about, say, for example, riboflavin, so vitamin B2. So we hear the word vitamin B2, we think of riboflavin, but it actually exists in multiple forms, much like the others. Some of those are active. So riboflavin becomes flavin mononucleotide, FMN, or riboflavin 5-phosphate is the other name. That's semi-active, that does actually have functions. And then there is the active form, which is FAD. So FAD is used in the TCA cycle in, say, succinate dehydrogenase. It's used in pyruvate dehydrogenase in parts in the complex. It's also in alpha-ketoglutarate dehydrogenase. So it's relevant to the TCA cycle and feeder, on primary feeder, and it's also used in the catabolic pathways, so things like the fatty acid oxidation and other places. So if again we're talking about the mitochondrial metabolism and electron transport chain before, the other feeders apart from complex 1, they're all riboflavin related. So when we're talking about a B vitamin deficiency, I think the concept then is it's not just the total amounts of the B vitamins that you're consuming, it's whether or not they're ending up in the active forms in a sufficient amount to be to be appropriate. And that's where it gets really tricky, because somebody that is, say, like a high performing athlete and who has a lot of oxidative stress and things like that, they might need a higher intake of B vitamins to support their mitochondrial load and metabolism. A chronic disease patient, much like the athlete, has the same issues, only more. So they've then got all of the rest of the systems that are actually dysregulating, and they've got, the mineral deficiencies, which are then contributing to the problems in the conversion of the inactive forms of the B vitamins and other things, and vitamin A and everything else, into the active forms. So one of the ways that we can try to compensate for that is by flooding the system with large amounts of the inactive forms, hoping to push some of these pathways along a bit to get enough of the active form and, in the case of, say, B3 and the others, to then go into a pool that can be also then, redox correctly. So it's correcting both the synthesis and the redox. So in the concept of the mineral terrain, so we're talking about metalloenzymes. So when we're talking about the proteome, there's about 26 to 2,700, it's still kind of being fleshed out, reactions, so enzymes, proteins, which require minerals as part of their catalytic and other activity. So, for example, if you've got an enzyme like lactate dehydrogenase, for example, we were talking about that before, it requires zinc. We're talking about the prolyl hydroxylases. It uses zinc as a stabilizer, but it also requires iron or silicon as the main mineral. And of course, it needs other things like oxygen and vitamin C as part of its overall reaction. The same thing happens with the glycolysis pathway. You might look at the fructose kinase reactions, PFK1 and 2, for example. They also require zinc. We might look at some of the top of that pathway. We might look at the glucokinase, hexokinase type stuff, which needs magnesium. Particularly important for glucose metabolism and glycogen metabolism is the magnesium. So in that context, if we were to look at the top of the pathway, if we have a magnesium deficiency, we're going to have significant issues with, one, creating glycogen, but two, all of the reactions that ultimately need ATP are largely Mg ATP or sometimes Zn ATP, or even in the context of cobalt in some reactions. So in the B6 metabolism, in some of the riboflavin metabolism, it has the option of using cobalt ions as part of the reaction. So these deficiencies that are affecting these metalloenzymes then have a knock-on effect for the actual metabolites being produced, which then have effects on the pathways, which then upset the homeostasis and then create the cycles of problems, the ripples throughout the cascade.
So let's talk then, since we're on the mineral conversation, about iron. So do you more commonly see low iron, or in some chronic illness circles, we talk about high iron from HFE leading into almost hemochromatosis, but not technically, that kind of over absorption and accumulation of iron that also causes too much of an issue? Or maybe we have too much iron in the blood, but too little in the tissues, and essentially have iron dysregulation. So what are your thoughts on iron? How do we bring regulation back to the iron conversation? How does then hepcidin fit into that conversation? And is hepcidin affecting more than just iron? Is it affecting other minerals? And thus, is it also leading to the accumulation of more heavy metals that we then have to focus on detoxifying more? So give us some insight on iron regulation, dysregulation, and then is there a role in your model for lactoferrin, which seems to be getting a bigger piece of the conversation recently?
Yeah, well, all very good questions. Okay, so they're all parts of the cascade in different places. So the iron regulation, the master regulator, as you said, was hepcidin. Hepcidin essentially is a peptide hormone that sits downstream of certain cytokine elevations and other things. There are a number of influences. But what happens is, let's say TNF alpha, so tumor necrosis factor alpha, IL-1 beta, 6, 10, 22, they're all promoters of hepcidin. The main ones are probably the TNF alpha, IL-1 beta, and 6, but the others are still relevant. And so when they're elevated, they promote hepcidin, and then hepcidin inhibits two metal transporters. So one of them is divalent metal transporter 1, and the other one is ferroportin. You can tell by their name. So ferroportin was initially studied for iron, divalent metal transporter 1, as you can see by the name, divalent metal. So it's already looking at a certain state of the ion or the metal or mineral, and ultimately it's able to transport quite a number of them. So there are 11 of them in the literature. Some of them are heavy metals. So you can look at cadmium and lead, for example. So then we move on to the context of how they're expressed in different cells. So if we were to look at the duodenum, it has both DMT1 and ferroportin. So what that means is that when hepcidin is elevated, it blocks the dietary absorption of all of those minerals, at least the ones that are using those transporters. And there are other transporters that will kind of sort of work, but it will block the absorption of the main ones. So iron and manganese are probably have the highest affinity for those transporters. So the biggest thing that we'll see at that point is that the iron and the manganese levels will go down. Others will also, be involved. Now, in other cells, they only have, out of the divalent metal transporter 1 and ferroportin, they'll only have one of those, which means that they have a different role. So when hepcidin goes up, they then continue to suck in the metals out of the serum and hold on to them. They sequester them until the inflammatory signaling and the hepcidin comes back down again. So that's part of this proposed immune response in an acute infection. That's basically the mechanism I see around that. The other effect with that then is that it's also pulling in the heavy metals. So the brain, the liver, the spleen, the kidneys are also then sucking in the cadmium and the lead at the same time as it's pulling in the iron and some of the other things. So it's an interesting cascade. So in the context of a chronic infection, like in this chronic disease spectrum, what we're seeing then is that over time, the dietary absorption of the minerals in a chronic, immune response state, inflammatory state, is then leading to progressive depletion. And the data is very, very clear on this. And I mean, hundreds and hundreds of panels that I've looked at that see this. But also we see the accumulation of the heavy metals, because even though the signaling has basically sucked it into the brain and the liver and the kidneys, if the glutathione metabolism is not working, it's now been sucked into the brain, the liver, and the kidneys. It's not getting kicked back out again after the inflammatory condition or the signaling has calmed down again. So that basically explains, I believe, in the model, how we end up. Okay, it gets described as anemia of inflammation in some language, in some articles and different parts of the medical sphere. However, the reality is that the iron is the canary in the coal mine. So if you're seeing the iron dysregulation, you're also seeing the same thing happening with the other minerals and the metals. Now we then talk about like the hemochromatosis, beta thalassemia type stuff, where we see the elevation of the iron instead. And iron, of course, exists in all these different states. We've got different redox states, Fe2, Fe3. It's able to be transported in the transferrin. It's got multiple different ways. It's complex, multiple different ways that we need to measure it. So when we start looking at the elevation of iron, then we can also talk about things like methemoglobinemia. So where we basically end up with an elevation of iron in a particular state, and then it has effects on the ability for respiration and other things. So In that context, if we were to look at things like cytochrome C activity, which basically is part of the electron transport chain, it basically is sitting there after complex 3, between that, complex 4, I think. As my brain off this morning, I might need another coffee. But it also happens to be handling the Fe2, Fe3 redox. And so you might end up, when you're looking at this, the overall picture of the iron, you might see an accumulation of iron in a particular form. And you will see, for example, the transferrin saturation sitting above its normal sort of set point of around about 40% in like a normal state. So something that I've seen is that if you look at those patterns, you'll see that those particular people also have the elevation of oxalates high, much higher than the others. They have a typically a higher heavy metal accumulation state as well. So you'll see like the elevation of the cadmium and some of the other things. You will see essentially a higher oxidative stress profile and an impaired iron redox. So they still have the inflammatory conditions, which are creating the dysregulation or the altered mineral metal transport, but they also then have excessive amounts of, and maybe there's some other differences that are affecting the iron metabolism redox to allow this accumulation in certain forms. So hypothetically, anecdotally, I've also seen that problem correct itself over and over again as these issues with the mitochondrial metabolism, oxidative stress, and the other things are actually resolved. So the dysregulation in the iron excess and accumulation, those patterns also resolve as the mitochondrial dysfunction and the quote unquote cooling system. So the glutathione redox and the other things are actually corrected.
So it sounds like there's not necessarily a need for lactoferrin.
Well, lactoferrin is in the protocol in the colostrum. So there is already an amount in there. It is useful, but it is helpful, but it's, as much as possible, if we can go upstream to correct the inflammatory state, then restore the normal absorption patterns and the metabolism, then great. There are multiple things in the protocol which work to address this while the bigger picture is being sorted out. So there's things like fulvic acid and humic acid, and there's other things in there which are all moving towards that. And of course, some of the direct interventions for the mitochondrial metabolism, glutathione redox, etc.
I mean, it's interesting that many microbes use iron to their benefit. Borrelia specifically uses manganese. You mentioned that when hepcidin is increased, that the minerals that then are most affected is iron and manganese. So I'm wondering, is it possible that the body increasing hepcidin is an intelligent adaptation to protect itself from infections?
That's exactly it. That's my take on it. It's an acute infection response. So it's meant to favor the host in an acute infection situation. But of course, if that progresses into a chronic infection because the body hasn't cleared out that acute infection, then it turns into a long-term potential problem. So it's a gamble against you beating up the invader early in the piece. But if that doesn't succeed, then it becomes an issue.
Okay, I'm going to ask a few more questions here and then we'll save the others for our next conversation. So, you talk about the importance of diet, the lymphatic system, stress. You've mentioned pacing and other lifestyle factors in your model. So, when you're thinking about diet, what dietary choices contribute to chronic illness? What choices are helping to lead us out of it? Are there any consistent patterns that have kind of led you to a somewhat healthy diet framework? I know you have your standard mixed diet that you talk about. What are the targets for the macros? Do you tend to prefer higher protein and fats and lower carbs, or do you find that some people do better with higher carbs? What foods are feeding our microbes the wrong inputs and leading to the production of more toxins in the body? We'll talk more about endotoxins and LPS. And then what are your thoughts on keto and carnivore in terms of your model?
That's actually the most complex question out of this whole series of questions for the last 2 hours. I'm going to do my best, but I don't think I'm going to cover the answer adequately, and maybe we can pick it up again on the next. A lot to unpack. So, okay, there are a range of things that happen because of the way that the body starts potentially reacting to a number of different dietary components. The behavior then becomes to avoid a number of these different inflammatory things. And some of that is very sensible. Some of that is unfortunately going to restrict our ability to obtain nutrients from diet, even if we are able to absorb them through that inflammatory cascade. So that's one of the challenges. The other part of this is the glucose and carbs and that relationship, and our ability to convert glucose into glycogen and to essentially, quote unquote, the insulin sensitivity aspects of it. So we have multiple things going on, and this is going to be I'll do my best to get through the answer in this. So we have essentially a need to maintain glycogen stores, and that need is progressively harder and harder and harder to fill because as the glutathione metabolism becomes more and more impaired, and the oxidative PPP, the pentose phosphate pathway, haven't really spoken about that, becomes progressively impaired, then the downstream regulation of things like the UGPase activity, which is required to regulate the glucose 1-phosphate into UDP glucose, which then goes towards glycogen, that becomes inhibited. So at that point, we start becoming glucose intolerant. So in other words, the insulin sensitivity is dropping. So therefore, we have more glucose in the system, which is not actually being utilized. And that means two things. One, it potentially becomes fuel for some of the microbial organisms in different tissues. It also then fuels, in this case, also when combined with oxalates, UTIs. So essentially we have oxalates, getting through the kidneys and into the bladder. The oxalates start creating damage to the surface. That creates an inflammatory condition, which allows the biofilms to stick. And we're getting glucose getting, secreted into the urine, which then feeds the microbes. Well, great. Well, we now have, biofilm colonies in the bladder and the kidneys and other places, which then lead to, all of the rest of the mess that we'll see. So there's that. So we kind of have a essentially metabolic syndrome slash this, essentially type 2, type 3 diabetes being described in the model. So we kind of need to still try to maintain the glycogen, but the system's not responding to large spikes in glucose very well. So then we need to progress to smaller meals progressively over the day, trying to maintain the glycogen without actually spiking the glucose levels. We then have the problems with histamine reactions. So for a variety of reasons, the histamine metabolism becomes dysregulated. So there's that, but look, and in the cell danger response and how that also applies. So the nervous system has a huge input on degranulating mast cells, particularly when the extracellular, ATP signaling is dysregulated. So people will react to different compounds in ingredients in food, not necessarily because they're somehow, like bad things. It's just literally that sure, there's barrier dysfunction. Sure, there might be, some of the immune response to things that are getting through the barrier, but the nervous system itself will start reacting to pH changes, to all kinds of different things when the calcium channels are dysregulated, which is, downstream of this sympathetic overdrive and other dysregulation. There's some more diagrams coming in soon and extensions to things like figure 18. Anyway, so the histamine response can then be driving and dysregulating some of the neurotransmitters, but also blood flow and everything else. Okay. The oxalates we've talked about a little bit before. So the dietary oxalates, we need to kind of, enough. So 50 to 100 milligrams if it's tolerable. Unfortunately, the excessive oxalates are going to do things like inhibiting the glycolysis pathway, messing up the thyroid hormone signaling, altering some of the metabolism around like the branched chain amino acids and things. So oxalate, as much as it's a problem when it's forming calcium oxalate crystals and things like that, and pain and everything else, it's also a regulator. So if we have a look at it, it's a signal that actually inhibits pyruvate kinase M2 at the same time as potentially inhibiting the enzyme which is creating lactic acid and 2-hydroxybutyric acid. So it essentially shifts the metabolism away from glycolysis and more towards branched chain amino acids. So there's some interesting sort of regulation downstream of that. The problem is, of course, all of these things work up until a point, and then when they get too excessive, it reaches a point where we've kind of destabilized the ability to restore homeostasis. So the regulation itself becomes, problematic. And then look, ultimately, we've got the foods that we're eating. They're not just for us; they're also to feed the microbiome. So if we start reacting to foods and start removing them, then we're going to no longer maintain the species of microbes that are needed to try to, maintain that balance. So it becomes a spiral towards, sort of oils, meat, keto carnivore type diets, trying to avoid the things which are triggering gut responses and inflammation and histamine to get a symptomatic benefit, but it traps you ultimately into a cycle where you can't go away from eating that type of diet because you're reacting to everything. So you can have it, you can exist, you can thrive even in a keto carnivore situation. I did keto for 8 months at one point for a number of reasons, but experimental data and just a general interest. And there was some really, really good things out of that actually. But ultimately, yeah, the getting glycolysis working is really important, not for brain metabolism alone, but also for immune regulation, right? The immune cells, they really, really like glycolysis and they really like glutaminolysis, a number of them. So getting working, hepatic gluconeogenesis and dietary intake of carbs and things restored is important for immune function.
Do you think that carnivore has the potential long term to reduce microbial diversity in the gut?
Yes. Look, there are studies are mixed on that, and you see the adaptations around that. One of the things that I've seen is that in a keto or slash keto carnivore environment, one study indicated that you would see a big shift in the microbiome for potentially up to 12 weeks. One of the things that I've suggested in heavy dysbiosis issues and heavy fermentation syndrome issues is a potential, and look, again, there are cons with it, period of an intervention period only, not a lifestyle change to keto, to basically cull the numbers of the fermenting species and then reintroduce, once the conditions are right, the carbohydrates again, to then start restoring some of the normal metabolism. And it's an unpleasant shift into keto. It's an unpleasant shift back out of keto as well. There's quite a number of things that adapt around that situation. And one of the things that you'll also see with a lot of people's data, when they're more severe, typically, you'll see that even though they're eating a mixed diet, they'll still be producing ketones. They can actually be in deep ketosis simply by the fact that their glycogen storage is so impaired. So it's interesting.
And I wasn't going to ask you this question, but when we're talking about optimizing metabolism, do you think that there will be a role for low dose microdosing of GLP-1s to improve metabolic function?
Oh boy. Okay, well, here's a fun question. So GLP-1s are a hack to basically produce more sympathetic flux. So in other words, if you've got problems with parasympathetic signaling and metabolism, glycogen storage, then it's a way that you can eke out some more TCA cycle flux coming from the sympathetic pathways by essentially mimicking the role of adrenaline without the adrenaline itself in affecting the CAMP-PKA pathways to basically drive more flux out of the catabolic pathways. So even on the microdosing, so there are big problems. If you start looking at the GLP-1s and lawsuits and black box warnings we're starting to get, one of the things you'll see is gastroparesis and suicide ideation and a number of other things. And I've unfortunately seen both of those, occur in people that have gone down that route. Unfortunately, also successfully in one case. So there was, yeah, it's something that essentially, if somebody's already heavily reliant on sympathetic signaling because their parasympathetic signaling and glycogen storage is impaired, then yeah, pushing further into the sympathetic pathway and bias is further away from the long-term goals of restoring the parasympathetic signaling and metabolism. So it's one of those things that you'll take it, you'll see if you're low dosing, and I definitely wouldn't do it at, and I can't give medical advice, but I would not be using the full doses of GLP-1s in pretty much any condition, my personal opinion. But if you were going to use a GLP-1, as soon as you get off the GLP-1, you go straight back into the same metabolic, problems where, you're not essentially able to produce collagen. And look, there are all sorts of other things there as well. So essentially, one of the things that you see is that you use a GLP-1, it then drives up gluconeogenesis. So your muscle tissue is essentially getting, eroded away to satisfy the requirements of the signaling. So it's not a free lunch. Yes, you'll lose a whole bunch of fat if you push it, but that's because you're essentially driving up the sympathetic pathways as a compensation for, the impaired other pathways, the glycogen.
So my last question for part one of our conversation before we leave the diet discussion, let's talk a little bit about the role of endotoxins, lipopolysaccharides, and how they contribute to these conditions. Is that a significant contributor? How much of a focus do we need to put on mitigating endotoxins to optimize health? And in your world or through your lens, when we're talking about endotoxins, are we talking about exogenous sources of endotoxins from building exposures like P-traps that have dried out or sewage backups, things of that nature? Are we talking about endotoxins that are being produced within our own microbiomes, or are we talking about a combination of both?
I think, look, if you were to look at the ratio of load from endotoxins from the environment versus our own microbiomes and barrier function issues, then I think, look, the environmental sources are a drop in the ocean compared to what we're normally experiencing every day. In that context, if you look at the LPS in the gut, under normal circumstances, there's a lot of it. Quite a number of the species are producing LPS. There's nothing abnormal about LPS other than when it gets through the barrier and ends up creating a number of, inflammatory cascade-related issues. So LPS in the brain does horrible things. LPS in other tissues does horrible things. Ultimately, we want to keep the LPS on the right side of the barrier where it can, live in a hostile microbiome and not actually cause us any problems. So the focus then needs to be more on the barrier function than it is the LPS itself. And sure, there are things like, spermidine and other things that we can use to, mitigate some of that. But the focus needs to be on why the barrier is actually, performing poorly in the first place.
I mean, it's definitely an interesting perspective, and I appreciate that because in the shoemaker realm, when we're talking about SIRS, endotoxins is now said to be 28% of the causation of SIRS based on their gene transcriptomics testing, but they only consider endotoxin exposure from the environment. They don't consider endotoxins that are being produced internally, endogenously in the body. And so that was always a question for me is like, well, how does the body know where it came from? Like either one potentially seems to be an issue.
I mean, there are different species of LPS as well. So just to bear that in mind, not all LPS is equal. But yeah, look, in the grand scheme of things, yeah, look, Richie's got a view on things, and that's great. I see things through a very different lens in many ways. So in the context of SIRS, for example, I'd be focusing on the same cascade that we're describing in all of this. So, Richie focuses on the HLAs. I don't see that as being a significant, variable at all. I think the genetic disposition towards some of these things is certainly present, but I don't think it drives the disease in any way. So I think essentially it colors the experience when you're actually stuck in a compromised metabolic state. So if you're already dealing with, quite a range of low minerals, if you've already got like the microbiome dysbiosis, if you've got the nervous system dysregulation, if you've got poor parasympathetic signaling and energy, localized energy storage, all of the rest of this mess is going to be more relevant. A 30%, reduction in, for example, the MTHFR metabolism based on genetics is likely to lead you more towards the, this methylation, glutathione, OXPPP axis issues, for example. But it doesn't drive it. Like I'm an example of that. I've got MTHFR and a bunch of other mutations. I need to do exactly zero to manage any of that. I don't need special supplements. I don't need anything, to manage any of these mutations at all. But, if I am in a compromised metabolic state, which hopefully I never will be again, then yeah, some of these things are more relevant to the symptoms that I might be experiencing, or it might influence my susceptibility to reach that state.
That's part 1 of my interview with Joshua Leisk. Don't miss part 2 in episode 234, where we discuss the immune system, structural issues, hypoxia, oxidative stress and inflammation, the mitochondria, mast cell activation syndrome, and the boron-free protocol. You won't want to miss it.
Thank you for listening to today's episode of the Better Health Guide podcast. You can learn more about today's guest, including relevant links and resources, in the show notes. This podcast may provide community discounts and occasionally share links from affiliate partners. You can find this and other episodes on YouTube, Spotify, Apple Podcasts, Amazon Music, iHeart, and more. To learn more about Scott's personal journey to better health, visit betterhealthguide.com. Join us next time on the Better Health Guide podcast.